Okay, so this video is going to be about the scientific methods that scientists use in order to solve a problem. And there are 6 steps in the scientific method. Identify a problem, gather some information, make a hypothesis, test that hypothesis, analyze the results, and then draw conclusions. We are going to look at a specific example to walk through these steps so that you can better understand them. Our example is pretty basic. It says, suppose you had 2 plants, a cactus and a palm. You planted them in soil and watered them daily. After 2 weeks, the cactus was dead. So here, this identify a problem is basically the same thing as a question, and we want to know, why did the cactus die? That is what we're trying to figure out. So we now have our problem, so we have to gather some information. So we want to ask ourselves, well, what do you know about these types of plants? Well, we know a lot already. Sometimes you don't have to do research, sometimes you do. We know that palm trees are native to tropical areas, and it rains a lot in tropical areas. We know that cactus are native to the desert, and it doesn't rain much in the desert. There will be times when you're actually going to need to do some research and get some more background knowledge. You just need enough to make an educated guess. You need enough information so that you can write a hypothesis that is a good, educated guess. When we get more into research, we'll talk about reliable resources. What is a reliable resource and what is not? Wikipedia is not considered a reliable resource. So now we need to say, well, how are we going to test our hypothesis? What does that actually look like? Oftentimes in a lab report, this is called your procedure section. So our hypothesis, or my hypothesis, is that I believe the cactus died because it got too much water. I say that because cactus usually live in the desert. It doesn't rain much in the desert. So I'm saying if it was watered daily, I think that it got too much water. So when we set up our test, we have to create some different groups. And since we're looking at the amount of water, did the amount of water kill the cactus, then what we want to change in our groups is the amount of water. So we're going to have groups that have different amounts of water. So I've set up these groups. Group A is going to be watered daily, just like what happened in the example. Group B will be watered weekly. Group C will be watered monthly. And then we're also going to have a control group. Your control group is the group that does not get any treatment. And in this case, treatment is water. So we have different amounts of water, and then the control group gets no water. Once we've set up our groups, we really need to make sure that we understand what our variables are. What's the independent and dependent variable? The independent variable is the thing that you are changing, often also referred to as the thing that is being tested. So we are testing the amount of water. So the independent variable is amount of water. The dependent variable is the thing that you're going to measure. What data are you actually going to collect? Now when you think about a cactus, there's a cactus here. When you think about a cactus doesn't really get taller, and it doesn't have leaves. So you can't measure the increase in height of a cactus, and you can't count the number of leaves. So when we talk about how we're going to measure the cactus, we want to look at maybe creating what is called a health scale. Now these scales can be created for all sorts of things. In this case, we're going to look at, does the plant look healthy or does it not look healthy? These scales do not have to be elaborate. In this case, I'm just doing a 1 to 4 scale, with 1 being a completely healthy cactus and 4 being a dead or nearly dead cactus. 2 would be for a cactus that's mostly green, but it is starting to show a few brown spots. And then number 3, if I were to give it a 3, it would be one that's mostly brown and it's starting to look unhealthy. And so this is how we are going to collect data. Every week on our groups, we are going to look and we're going to assess the cactus in those groups to see, do they look healthy or do they not look healthy. We're going to assign them one of these numbers, a 1, 2, 3, or a 4. Sample size is something that we will talk a lot about as we move through the year, especially when we get to science fair. It's really important that you have a large enough sample size that you can be confident in your results. So here you would say, well, let's just put one cactus in each group. Well, that's not going to be enough. One cactus is not going to be enough to give us good, reliable results. We are going to say we're going to put 10 in each group. If we just put one in each group and there was something wrong with that cactus, we would never know it. If we put 10 in a group and there happens and 9 of them do exactly the same thing, but one of them is completely different, then we know that there was probably something wrong with that cactus and we're going to throw it out. We're going to call it an outlier. So other than our independent and dependent variables, we also need to consider what are the constants in our experiment, the things that are going to be exactly the same for our 40 plants. So you say 40 plants, what are you talking about? So if you go back here and you look, Group A is going to have 10 plants, Group B, 10 plants, Group C, 10 plants, and then there will also be 10 in the control group. So there is our 40 plants. So with our 40 plants, the only thing that's going to be different between the groups is the amount of water. So we have to think about what are all the other things that plants are going to need, and we have to make sure that all of those things are the same. And those things are what we call our constants. They remain constant. And so we know plants need sunlight. So we have to make sure that all 40 of our plants get the same amount of sunlight. We also need to make sure that all 40 plants get the same soil. They need to have the same type of soil, and they need to have the same amount of soil. If they were to have more or less soil, they may do better or worse depending on the soil, and we don't want that to be the case. We want to know if there's a difference in their health, that it's strictly because of the amount of water that they're getting. They all need to be in the same size pot and the same type of pot. And these pots, we need to make sure that we label them. So each group has 10 plants. So one plant is going to be labeled A1, A2, A3, all the way to A10. And we'll do that for the B group as well, and also for the C group. And then we will also do for our control group, and we can call those X1, X2. So we need to label all of our plants so that we can so we know which plant is which from week to week. We need to make sure that all of our plants are in the same temperature. We can't have some of them being cold and some of them being hot. They all need to be in the same temperature. We need to water them all at the same time. They'll be getting different amounts of water, but we need to make sure that we water them at the same time. If we're going to water them in the morning, we water all 40 in the morning. Or if we choose to do it at night, we water all of them at night, but we always water them at the same time of day. Same thing for doing our health assessment. We want to measure them at the same time of day. We're going to do this at the end of each week. You don't need to do this sort of thing daily. You can do it weekly. So at the end of each week, we would look at all 40 of them, and we would assess all 40 of them at the exact same time. So here's some sample data. We call this raw data, which means that it's all the data that you collect. And if you look here, we are going to collect data for 10 weeks. And every plant, this is group A, and this is plant 1, plant 2, plant 3, plant 4, and so on. And so week 1, after 1 week, the number 1 plant was still looking really healthy. These were starting to get a little bit of brown, but for the most part, after the first week, they all looked really healthy. This column right here shows you the average of these 10 plants. So 1, 2, 2, 1, 1, 2, 1, 1, 2, 1. All of those average to be a 1. So that's how you read a data table like this. If we look at the raw data for group B, it's set up exactly the same way. We have our 10 weeks, and then we look over here at the average. Now, if we go back, we can see that this is Group A. Group A is the one that was getting watered daily. So the ones that are getting watered daily, after the second week, they died off pretty much completely. If we look here at Group B, these are the ones that are getting watered weekly. Well, they started to deteriorate around about week 5 or week 6, and they really only started to die off about week 8 or 9. So what you do with raw data is you have to analyze it. You have to say, well, what happened in general? And so this is where you're going to show all of the averages. So this is the average health score for each group by week. And if we look at this, we can see obviously Group A did the worst. Group B, probably the second worst. Group C, this is our group that only got watered monthly, did really, really well. They stayed healthy the entire time. And then our control group actually stayed healthy until about the 7th week. But then they started to slowly lose their health. So to analyze the data, we want to put it all into a data table, and then we also want to write it down in words. What happened? So in this case, we just analyzed for each group. Plants in Group A died the fastest. Plants in Group B died the second fastest. Plants in Group C remained healthy for the entire 10 weeks. And plants in the control group never died. But they started to become unhealthy after 10 weeks. So once we have analyzed the data, this is where we go back and we try to answer our question or write an answer to our problem. So we wanted to know why the cactus died. And our hypothesis, we said that the cactus died because it got too much water. So in your conclusion, you're going to need to revisit your hypothesis. So here I say, in my hypothesis, I stated that the cactus died because it got too much water. And here, the data supports my hypothesis. We are never going to say that my hypothesis is correct or incorrect. The data either supports it or it does not support it. So here the data supports my hypothesis because the cacti were watered daily, because the ones that were watered daily died after a week, and the cacti that were watered weekly slowly deteriorated and then died after 9 weeks. The cacti that received water on a monthly basis stayed healthy throughout the experiment, showing that cacti don't require much water. So here I am. Experiment showing that cacti don't require much water. So I'm saying these are my results, that I think the cactus died because it got too much water, and the data does support that. I further go on to sort of talk about maybe why these results happen, talk a little bit about a cactus plant. So this is probably why they are prevalent in the desert. The control group actually did not die after receiving no water for 10 weeks. They did, however, start to become slightly brown and shriveled. So I'm going to make a prediction. I predict they would recover quickly if given a small amount of water. Living in the desert, they are probably accustomed to going long periods without water. So this is the scientific methods. We will talk about this in class a lot this year. And if you have listened this far, you should know that my mother's nickname is Squeak.